useful. We just have a new participant. Okay, I think everybody is here. Marvelous. Okay. So what are we talking about? Basically, I just wanted to go over what the UK Biobank is, and then what this paper particularly is reporting on. Some of you may be more or less familiar with the UK Biobank. It is an absolutely phenomenal project, just an unbelievably ambitious project. Started probably almost 10 years ago, I think, at this point, maybe closer to 6 or 7. But this is a sort of science news blog where they were basically pointing out it's making the results available to everybody, which is really the critical part. So it's a prospective study looking at genotypic and phenotypic data on 500,000 people from across the UK who were aged between 40 and 69. And it is, of course, an open resource. You can get all of the data, the sociodemographic, the neuroimaging, cognitive tests, the health records, physical activity monitoring, x-rays, body imaging, so not just the brain, but the rest of the body, all sorts of measurements of the physiology, lifestyle factors. You'll see that in some of what they were looking at, as well as the genotypic data, which keeps rolling out, and of course then getting pulled back because they have to do quality assurance on it, and then rolling out again. And they're rolling things out in stages. So first there were 10,000 data sets, and there were 20,000 data sets. This is actually looking at all 400 and 1,000 data sets, because again, not everybody has everything. Most people have most things. This is a phenomenally ambitious project where they are actually built scanning centers. They actually built special scanning centers that could basically scan almost 24/7 at 3 or 4 sites in the UK, and then just recruited people as they could get them, that actually, the folks who actually fit. They tried to make the criteria as broad as possible, as long as it was safe for you to be scanned. And I think as long as you didn't have, I forget exactly what the rules were, but you were not supposed to necessarily be a healthy, super healthy control. They were looking for people who were the broad range of the population, and then collecting all sorts of things and following them for 10 or 20 years to see, can we predict who is going to age in a healthy manner, and what kinds of factors, either genotypic or physiological or economic or environmental, really make a difference in predicting some trajectory into old age. So this is not a developmental study. There's nobody under the age of 40 in it, and most of them, skew older towards the 50s and up. But they are making it publicly available, so anybody who wants to can go get it, which is always fun. You can basically ask for it, and then you can download the data. Now, it's a little bit challenging if you're slathering at the idea of actually leaping on this kind of data. Just be aware that if you can't necessarily collaborate with somebody else who has the UK, if you both have the UK Biobank data, you may have gotten it anonymized in different ways, because they are very, very protective of it, and so they don't want someone to download it, get it, and then share it broadly with no protections. So they tend to anonymize it each time it is shared, so that one person's analysis can't be shared with another person's analysis, unless you deliberately go in and say, from the beginning, we're a team who are going to be working together on this data, in which case they get a common data access. So you really have to plan ahead when you want to work on the UK Biobank data, but nonetheless, it's not that hard to get. And it's an ongoing dynamic project. Looking it up recently, I realized that last year they added in COVID-19 testing to see if what can they predict about the long haul approaches, or who's going to be more likely to catch it, etc. So they're actually pulling that into the repository as well. So hey, you'll be able to check on the effect of COVID-19 on the brain in these people as they age as well, for those who, for the 1 or 2% or however many it was who actually developed COVID-19 or were asymptomatic, hard to tell. Now, there's also lots and lots of genetic analyses. As soon as they started rolling any of the genetic data out, papers started coming out, because people were just slathering over this data. and leaping right on it, starting with looking at smoking, lung function, chronic obstructive pulmonary disease, the effects of on body mass index, right?
Can I add one thing about COVID? There
Yeah,
are a
sure.
lot of cases, dementia, having
Yeah.
associated dementia. So it's not something that, it's been demonstrated that people have indeed vascular problems, a very small percentage, but indeed there's COVID associated dementia.
Yeah, yeah. So then that data is going to be available as well. So it's
Yes.
really, really lovely. In a sense that they're making lemonade out of those lemons. So there've been basically genetic analysis after genetic analysis, looking at everything, including stuff you would not think was necessarily genetically related, including can you predict who's going to participate in the optional components of the study? It turns out, yes, with enough data subjects, yes, you can actually try to figure out who is genetically predisposed to volunteer for additional testing. You can look at cognitive functions, physical and mental health in the UK Biobank. And of course, this is one that Nora and I, working together with Vince and David and Amanda, Amanda did much of the heavy lifting on this, using the UK Biobank to look at genetic contributions to some of these gray matter patterns and what that means as well. So you can, and this is only a subset of the genetic papers that have been coming out on this. And that really is what drove, I think, in many cases, this paper to say, let's just pull it all together. Let's, instead of everybody doing their own imputation and their own quality assurance processing and their own protocols, let's just run it once on everything, using the same processing techniques and the same genetic models and everything else, so that we can actually do comparisons across all of the phenotypes that have been collected in the UK Biobank. So here they've got basically 118 non-binary and 660 binary traits from slightly less than 500,000, because as you know, you start losing subjects as soon as you start doing any sort of quality assurance. And then they've compiled it into a publicly accessible database so that you can actually look at their association effects and get their summary statistics for the various genetic variants. So you can actually query the UK Biobank, which is lovely. So let's see how they did it and what they're actually finding. So let's talk a little bit first about the sociodemographics and other characteristics of the UK Biobank. Because it was intended, of course, to be something of an epidemiological sample. It was supposed to be representative of the UK. And it turns out, of course, it isn't. There is a certain amount of what they're pointing out here of this healthy volunteer selection bias. You tended to have basically a healthier population are the ones who were volunteering for this study. It tends to be slightly, they tend to be slightly older. Obviously, that's sort of part of their, they're looking for the older population, but also that more women are showing up to volunteer for this study, and to obviously be more well-to-do socioeconomically. They're less likely to be obese, less likely to smoke, less likely to drink alcohol. So they're, even though they didn't recruit for a healthy population, that who has the time and capabilities and interest in participating in this study. So you have to keep that in mind. This is largely a healthy population, but there is a range. So you're not. I was originally thinking, my goodness, if they've got 1 to 2% of 500,000 people have schizophrenia, we are gold. This is amazing. But nobody with schizophrenia signed up for this study. Now they do have lots of psychiatric conditions. There are lots of people with anxiety, depression, or a history of depressive episodes. There are some psychiatric, measurements that are included, but for those of us particularly interested in psychosis, this is not necessarily a panacea, unfortunately. But there's lots. It's amazing what you can actually find out from it. So things that we should probably be aware of and make sure we understand as we're trying to work with this resource, the GeneAtlas resource, is the difference between binary and non-binary traits. They toss around the idea of Snip heritability a lot that we should sort of dig into a little bit. The HLA area is that they talk about genetically is extremely important, and it's worth knowing a little bit more about that. And then what can you actually do with the GeneAtlas database? So talking first about the binary phenotypes, that's just basically, are they present or absent? So, so for example, this is a number of those phenotypes that were binary. So do you have a chronic lower respiratory disease? Yes or no? Not which one is it? Not how long have you had it? Not any, not what effect does it have on your function? Nothing like that. Just yes or no. Do you have it? Do you not have it? For example, Parkinson's disease, do you have it? Do you not have it? Not what stage are you in? None of these really detailed pieces of information. But do you have it or not? Now, because the medical records are also attached to these people, you could then go in and say, okay, let's look at the people who say, yes, they have Parkinson's disease, and then try to dig out other information. But it's an informatics challenge, let's say the best, but you can certainly do it. Then there is non-binary phenotypes, which is a range of values. So this is an, again, example of some of the non-binary phenotypes. And it gives you an idea of what kinds of phenotypes they were asking about, just a range. There's things like, okay, in your blood, all sorts of reticulocyte information from the blood, handedness, how strongly handed are you? Not just yes or no, are you right-handed, left-handed, but which direction is it? How big were you at when you were 10 years old? Somehow they turned smoking status into, maybe how much do you actually smoke? But then there are things like, how much cheese do you eat? How fast do you usually walk? How many vehicles do you have in your household? What is your skin color? How much pork do you eat? How hot do you take your tea? I mean, they're just asking all sorts of phenotypes that, I wouldn't necessarily think to ask in a medical establishment, but they're basically trying to cast a very broad net and a very deep phenotyping as well to say how much can we actually find out about these people, and what difference does it make? Like how much oily fish versus non-oily fish? It's just unbelievable, the questions that they went into. It's tremendous, the data that's actually there. All right, so so that's the difference between binary and non-binary, and one of them is a continuous variable, one of them is just presence, absence. Genome-wide association methods, they go into a great deal of detail about how the people were actually genotyped, how many SNPs there were, the imputation techniques that they were using. For the purposes of GeneAtlas, they used a very specific imputation method and made sure it was the same for all of them. And then what steps they went through in the quality assurance. And then, of course, how do you actually do the analysis between the genotype and the trait? For those of you maybe not so familiar with how quality assurance works on the genetic data, clearly they wanted to make sure that each person's genetic data was sufficiently complete that you could trust the remainder of the data. Because even with the best genotyping in the world, they're 99.999% complete, but you've genotyped 5 million data sets. So there's always going to be 5 million SNPs. So there's always going to be a few SNPs missing for every person. But are they missing too many? Are they missing in a way that indicates something went wrong with the genotyping? You want to make sure that you've sort of removed all of that. And then they particularly said, we don't want really rare SNPs. Even with 500,000 people in the data set, they wanted to focus on SNPs where the minor allele showed up at least 5% of the time. So that's that trimmed things down a little bit, but you want to be clear on exactly how they did it, because that affects, again, your interpretation of what you're finding. If for example you're really interested in a rare allele, they might not have looked at it, or if you end up doing a sort of a linkage disequilibrium clumping on your data set and get a tagging SNP, you're going to want to dig into that a lot, because they may not have found the same tagging SNP. They may have done it slightly differently, etc. So there's, you definitely, when you use the GeneAtlas, you want to have in the back of your head, what are the differences between their genetic approach and anything else I'm looking at, either my own data or other papers that I want to compare to, etc. So their analysis technique was a linear mixed model. Those of you who have seen linear mixed models in your stats class, it should be fairly familiar to you. It's kind of nice, you get used a lot. Basically, Y is your dependent variable, right, which is your phenotype over your 500,000 people. It's a vector. X is your fixed effects variables. Sex array, the array that was used to do the genotyping, the testing site where the person was actually recruited and scanned and everything else. Age squared, because they're in that 40 to 80 range where things are going to be actually changing quite a bit. So they include a quadratic term. And then they also try to account for any sort of hidden genetic structure by taking the first 20 principal components analyses of the genome, which of course reflects your ethnic background and where you're from and everything else, in a way that you can get from the genome that you can't necessarily get from asking somebody. Then of course beta is their estimated effect sizes for all of those fixed effects, and then the meat of the matter is the G matrix, which is all of their genotypes with a minor allele frequency greater than 5% that pass their quality assurance of, making sure that they were available, they were, SNPs that were high quality, etc. And then the question, of course, then the noise term is assumed to be normally distributed. And again, when you're looking at their results, you want to take this into account. They were using a specific linear mixed model. It's highly defensible, but other people may have done it differently when you're trying to compare results across different sources, resources, studies, reports, or your own approaches. You may say, you know what, I don't think these are linear results. Maybe I want to look at some other analysis. Is that going to change what I see in the results? But for the purpose, for the moment, this is absolutely fine. And this is, generally speaking, how it's done. If you decide to do something else, you would definitely have to defend yourself in terms of why you would be doing it that way and what it would give you that the linear mixed model doesn't. Now, how did they decide what was a significant genetic association? For the purposes of most things, they said, look, anytime somebody does a genome-wide association study, generally speaking, we do a 10 to the minus eighth, because that was the argument 15 years ago. The 10 to the minus eighth was good enough for capturing what is specifically related to an individual SNP, given what we know about the correlated distribution of signal and independence and, the number of genes, and et cetera, et cetera. 10 to the minus eighth is sort of how we came to it. So they present a lot of things at 10 to the minus eighth, but then they also will vary their threshold. And in the GeneAtlas webpage, you'll see they allow you to actually vary the threshold so that you can explore perhaps more significant, more stronger effects or weaker effects if that's what you're interested in. What is SNP heritability? Now, I know some of you have been to workshops on what SNP heritability is. It shows up a lot in different genome-wide association studies, and it's a way of looking at the global effects of the genome without being interested in any specific SNP. So the technical definition is that SNP heritability basically measures the proportion of the phenotypic variance, so how much variation in the trait you're actually measuring, is really explained by the contribution from all the measured SNPs that you've got, not just any one, not just one that may be significant or not, all of them. If you know all of the measured SNPs and you look at all of the genotypes, how much of the phenotypic variance is accounted for by that. And there are lots and lots of different ways of doing it. They chose a particular way of measuring SNP heritability, again, as a standard technique for measuring it. But this is just an interesting way of looking at it. And a key point from the paper is, of course, the heritability of a phenotype can range from 0 to 1. It will never be 1, generally speaking, except for Mendelian disorders. We tend not to get a heritability of 1. So most of the time, there's variability in the proportion of the phenotypic variance that can be actually explained. And then this is sort of looking at the number of phenotypes. You'll see there's 1 or 2 phenotypes out here that with very, very high SNP heritability. That's generally body mass and height, tend to be the ones where if you know the person's genome, you've got a pretty good sense of their body mass and height. There may be a few others out here, I don't remember. And then it drops off rather rapidly. And of course, most of the phenotypes have very, very small heritability. So even if you know the entire genome, you don't have a lot of the variability of the phenotype. And then they basically were saying, look, under what conditions are they actually significantly different from 0? And you'll see down here in the lower heritabilities there. You've got more where it's not significantly different from zero, but you've got, you still have some where the fraction of the heritability can actually still be significantly different from zero, depending on what the variability actually is, and that this might be a significant proportion of the variability in the phenotype to be able to explain. It just depends on the actual phenotype. But basically, most of them very small. And the amount of cheese that you eat, the number of vehicles in your home are probably down around here, but I don't know that. You can explore that. Again, if anybody has questions, please just break in because I'm not watching the chat window or anything else. I did want to bring up this paper. It's a very recent paper from computational and structural biotechnology that's actually talking about lots and lots of different ways of estimating Snip heritability, because I've only learned about one or two ways of estimating Snip heritability, and it turns out it's its own field. So knowing how to estimate Snip heritability, it's well worth actually taking a look at all of these different approaches and why they might be more or less applicable to any particular question that you're asking or the phenotype that you're interested in. And once they were able to decide, they decided how they were going to calculate Snip heritability. The nice thing that they did was they could say, look, for many of our phenotypes, such as hearing loss, weight, colon cancer, height, breast neoplasms, rheumatoid arthritis, etc., there are already large and well understood and well regarded publications of the estimated Snip heritability for that trait. And so we can go and do our own analysis. And how does our analysis compare to their analysis? And generally speaking, it's pretty darn close. So they did a really nice job of basically saying our techniques are not going off in some random direction. We're well in keeping with what we already know in the literature. So basically, we're doing this right. We're doing this in a way that the rest of the research community can understand, and you can sort of build on it and compare results that way. So I thought that this is one of their supplementary tables that I thought was a nice addition to include. Actually, so body mass index and height are still only at like 0.5 and 0.25. I mean, they're not great. I don't know what the 0.8 one is. I'd have to go see. It'd be interesting. And also, the more heritable the trait actually is, the more significant associations there tend to be across the genome, certainly for the continuous or non-binary traits. Basically, the more heritable it is, the more loci on the genome are actually significantly associated with that phenotype. Binary, a little bit less so. But you still something of an upward range to say, okay, even if it's really heritable, how many significant SNPs do you have? Surprisingly, not a lot, which has possibly some interesting interpretations.
Can I mention something? This is fantastic. Thank you so much for putting all this information. I just want to say that point two, it was, when you have a schizophrenia patients and you do the study only in schizophrenia, using this Snip Heritability is point two as well.
Yeah.
So it's not.
No, I think isn't it smaller than that? Isn't it like 2%? Where this would be like 20%.
No, point two is the value for the Snip Heritability from the PGC, for the PGC.
Yeah.
Yeah. So
schizophrenia is basically right in here.
Yeah, exactly.
And that's one where that there's a genetic component. It's just
there's a genetic component. You have the patients.
You have your families. You can track it through.
Yeah, and that's where you get it. You get it at point two.
It's one of these it's part of what makes genetics such a gripping field, is saying, okay, how does this all work? Let me say a little bit about the HLA area. This is the human leukocyte antigen. I'm more used to calling it the MHC for the histocompatibility complex. It's on chromosome 6. These HLA genes are highly polymorphic. They're active in the immune system response, and they have many different alleles. And it's a really busy spot for increased risk for all sorts of things. The top hits for schizophrenia were found in the MHC complex, and the top hits for a lot of things are found in the MHC complex. And it is one of the areas which they specifically enriched in their genotyping because they knew it was going to be an active area, and they were not wrong. You can see this is the HCLA area, the HLA area on chromosome 6, and it's the one that has massive numbers of associations, mostly in the present absent area, but also in the non-binary areas. It basically, if you look across the genome, a priori, the associations with any phenotype, and the UK Biobank has hundreds of phenotypes, it's not uniform. Basically, there's an a priori expectation that if there is anything, it's going to be in the HLA area. But there are lots of other areas as well that seem to be enriched for associations with disease, either binary or non-binary. And I thought that was just interesting because everybody was making a big deal out, oh, wow, we get this stuff in the MHC. And it's like, well, but they're not the only ones. It's not specific to that disorder. It's really related to it. It has relationships with a surprising number of things that can go wrong. And that's another research point I think people will be or have been digging into and probably will be continuing for a while. What is it about this area in chromosome 6? The covariates, all of these covariates like age squared, gender, no, they didn't do it, no, not gender, but age squared, the site that they were in, oh, maybe it was sex as well, are actually important. So predicting, they did a whole bunch of trying to predict the phenotype, either present absence or trying to predict the actual value of the non-binary trait. It's better when the covariates are included. Generally speaking, this one was a very nice figure, which shows basically, again, looking at your SNP predictability from 0 out to about 0.5 or 0.8. And this is the correlation, right? So this is sort of the what is it that you actually predict, and what is the correlation? So SNPs, phenotypes that have greater heritability, SNP-wise heritability, they do a much better job. Basically, it's a very nice correlation in predicting from the genetic markers what the actual phenotype is going to be. If you include all of those covariates. If you don't include the covariates, you can see how it falls off rather tremendously. Basically, they're basically nicely correlated up until about 0.5, and then your ability to predict your phenotypes or flattens out at about point. They can't do a better correlation than about 0.4. And with the binary traits, it's similar. Basically, you can't do a correlation, but you can do an area under the curve, which again sort of gets at a prediction of case control status, if you will, or 0, 1, presence, absence of the phenotype. And, they're basically, that measurement is increasing with the heritability of the phenotype, as long as you include all of the covariates. If the minute you take out all those important covariates about the actual array that they were genotyped on and everything else, it flattens out at not so good. But it's, it basically, there's the best it can do, and then there's a couple that work without the covariates, but mostly you really need to know where were they genotyped, how was it done, all that good stuff as well, which is something to keep in mind.
Can I ask why the covariate of age would impact this at all?
Oh, I think it's largely because of the age relationships with most of the phenotypes.
Oh, okay. Okay.
I would imagine, they want to take that into account. So making it all searchable, right? This is the GeneAtlas, and this is under the frequently asked questions. And I strongly recommend you start with a frequently asked question so that if you use it, you know how to cite it. And it explains all of their terms when they say effect allele, when they say what the units are, etc. They really do explain it. And I wanted to actually go to the website and sort of walk through it a little bit. So this is first the paper on Nature Genetics, and I was going to say you definitely want to get the supplementary text, etc., because in The paper itself is, of course, sadly brief, and they have to put a lot of things into the supplement. So you want to make sure that you actually get that and refer to it and try to understand what they're showing and why they chose those tables. And as it is so usually true for such a large study, they have lots and lots of supplementary tables, some of which are just, mild variations on each other, but to get, just to cover the whole approach, all the possible approaches and all the possible alternate interpretations. All right, so let's actually look at the GeneAtlas here and play with it a little bit. So this is all of the individuals who were included, all of the traits that were included, and the number of variants that were included. And I thought 5 million was as much as we wanted, or 9 million would be as much as we wanted to. But they're doing all 30 million. And of course, they had to do this on a large supercomputer because trying to actually develop both the empirical or basically doing some of the simulations that they needed to get their p-values and whatnot were very interesting and very, very could take weeks.
We're
But
still looking at the how to cite the GeneAtlas.
Why? Okay, let me stop sharing that. Close that. Are you still, or what are you seeing now? You're still seeing the PowerPoint. No, stop the application. Let me actually share my screen. Here we go. How's that?
That looks good.
Okay, thank you. Alrighty, so this is the GeneAtlas and all of the relevant information. I rather like this design because they put the links at the bottom as well as at the top, because many of their pages you end up down at the bottom and then you want to go to the next thing. So you can get all sorts of information about exactly what the traits were that they used and how to get information about them. So it'll tell you, for example, what did they mean and what was the what are the fields in the data if you've downloaded the data, and then what were the genetic effects that they were finding, and all the different ways that they had of looking at it, as well as the genetic QQ plot to say it probably enriched for some sort of genetic effect, but it's, let's just say, not whopping. A little something near, a few things over here. And let's see, what else did they have in here for take out the Parkinson's? They had other, so they have psychological and psychiatric problem. It is not well defined. It's basically lots and anything from anxiety, phobia, OCD, depression, major depression. I forget what else they've got in there. Any sort of psychotic episode with or without a diagnosis of schizophrenia or bipolar disorder. I think bipolar disorder is actually in here. Basically anything that would be a psychiatric disturbance is all thrown in together. And then they're basically saying, well, if you use just the 623 that we think are relevant to psychiatric issues, you can see a little bit in chromosome 6, again, your HLA, and then a little bit on chromosome 7. And if you start using your 9 million, again, same areas. And if you do the imputed, actually there's, a couple things on 1 and 10 and 18, and you can drill down and see what's actually going on. Again, this is not specific to a particular psychiatric problem. It's just generically the likelihood of developing something is included here. So you can play around and get information sort of overall about the traits. You can then also search by variant if you know exactly what it is that you want to look for. You can search by region or gene. So you could basically say, oh, show me what's going on in chromosome 6 vis-a-vis bacterial infections and psychological infections, and see what happens. Do a plot because that's more fun. And you get all sorts of, basically in the bacterial results, there's not a whole lot going on. Psychiatric results, you can see a certain amount of things going on, and then you can dig into it. And of course you can also download all sorts of fun things, which God knows you probably don't want to do, but you might want to look at the sort of do a polygenic risk score for something, and see how that compares. You can look at regions of genes or phenome-wide association studies. So you could actually pick a variant and then see what's actually there. Let's take an example. And let's look at a plot because I like plots. And There you go. So then you can actually see, okay, for this RS, where, you know, from all the traits from 0 to 780, there's actually quite a bit of over here. And these are all, you can sort of see, has to do with the amount of fat in your body, arms, legs, etc. Is that basically across all of the phenotypes, you can, this particular variant seems to have something of an effect on those phenotypes. So that's actually, you know, sort of the fun part of it, to explore and see where these different things are. So disorders of the schizophrenia, disorders of the brain, things like that. Dun, dun, dun. That's all in this area. You can look for, you know, what seems to be related to these things. Oh, hey, look at this. Diabetes and body fat. Surprise. So that's the kind of rabbit holes you can end up going down. But I wanted to encourage everybody, particularly to, you know, to go and look at it, to look at the phenomes that are available, to look at the traits that are available, to, if there are, for those of us looking at QTLs where we have sort of prescribed areas of the genome that we're interested in, we could certainly look there to say, what did the UK Biobank find? If you have a SNP that you're interested in, you can certainly look at the UK Biobank to say, anything show up for that SNP, you know, just type it in here and see if anything shows up. So it's, again, another resource to use to try to understand the SNP related genetic effects. Doesn't, you know, at the moment, they are trying to look at other issues like methylation, etc. I don't know if those data are available. They're certainly not in a resource like this just yet. So that, I think that's everything I had to say. Are there questions, comments, updates, thoughts? Just look and see if I had anything else I wanted to point out.
So I have a comment that I've been reading. You know, I'm the diversity director for neuroscience.
Oh my.
Don't ask me how well.
How did that happen?
One of the things we had, and that was in the World Congress of Psychiatric Genetics, the idea that they, like you said, the data is biased towards Caucasians.
Absolutely.
Really. They have 10% of, you know, non-Caucasians, they haven't analyzed those.
Wow.
So any data that you get from there, you have to really think about what is the population they analyze.
Yeah.
And this is something that happens in most, because they are volunteers, like you said. The people that work all the time are not going to be able to go there and volunteer. So it's more, it's invaluable. I think it's really important to look at that in terms of many disorders, with the caveat that, yes, it's more a selected population. And like you said, it's healthy population, though they have SNPs that are associated, you know, in the population. We have a lot of schizophrenia associated SNPs. You don't have a schizophrenia unless you have the vast majority. And again, the other thing I wanted to comment is, you know, when you have a 2.2 heritability, why does it matter? Because it's only, they are only looking at SNPs with 5% or more, the minor frequency and the minor allele. So, like you said, methylation, there's a lot of epigenetic regulation and also rare alleles that they are not analyzing. So it only gives you a more global picture without going into, you know, details. But it's amazing that they have done, you know, fantastic job.
It's tremendous, absolutely tremendous. And I was wondering if it might be worth, I wonder if they're planning to run the 0.01 rather than 0.05, because it seems like with 500,000 people, you could take it down to 0.01 and still get some reasonable variation. It's not like everybody will have the same allele.
Yeah, that would be better.
But with speaking of the all Caucasians, I did actually want to share. I was presenting last week to so high school, classes that want a science class that wanted to know about genetics of schizophrenia. And I was talking to them about this paper that just came out last year. Did you see this one, Nora?
No, no.
This
Oh,
one's really, really spiffy. Because
oh,
they,
I don't know how I missed that. Thank you.
yeah, it's really cool. Molecular psychiatry last year. So they didn't get, what they did was they actually got a whole lot of Latino, South Asians, African Americans, and they did the genotyping to show, okay, yeah, they're actually different. And then they did the GWAS on them. Now they're smaller samples, so they managed to get 10,000 African American samples, and 4,000 Latino samples. But then they could compare it to the PGC, which is of course largely Caucasian. And so they could show that it was largely the same genetic loci, but it was often a different SNP that seemed to be the key player in these different genetic backgrounds. So it would be in the same area, maybe even the same gene, but definitely not the same SNP. And then a few, particularly in the Latino case, there was actually a whole new loci identified that did not show up in the Caucasians or the African Americans. So they basically were arguing there are common genetic effects, but there are also genetic effects that are specific to the different ethnic backgrounds. And what I really liked was this one where they did the polygenic risk score. So they had a polygenic risk score based on the African American analyses, the Latino analyses, obviously the PGC, and then they put it together to do a trans ancestry polygenic risk score, and they could basically show, the ethnically specific ones worked very well, but if you really want to do everybody, you got to put them all together, and then they could actually do a reasonable job across all the backgrounds if they actually use their risk scores, that combined everybody as opposed to the PGC. So I really like that.
Thank you for doing this, to pointing out. Yeah, I've been trying to do that. Unfortunately, I didn't see this paper when I did a presentation in January, except to say that they needed to include all these different populations because the SNP, the minor allele frequency is very different.
Yeah, it can be different. So it's very, I thought it was very promising. The teacher of the class was like, why are they only just starting to talk about African Americans in 2020? And I said, let me explain that. This is difficult. But I thought that, the overall sort of take home message is if you really have to put everything together to get your best possible, particularly if you're going to go for individualized medicine, any sort of precision predictions. You don't want to just use the PGC, willy-nilly across everybody. And I think that's certainly in keeping with what we've been seeing in our schizophrenia groups as well, is that, depending on the ethnic breakdown of the groups, you can get very different results.
Yeah, you had that recently. I don't know who presented that, when you combine different populations, you have different results.
Yeah.
Yeah. Wow, thank you. This is awesome. Thanks so much.
Are there questions or comments from other folks?
Hey, Jess, this is Jean. I mean, that is really rich information from this group. They did this. I'm just thinking in terms of, they did this work and how we can leverage the result to help our own study, right? So right now, I think we are thinking about using biobank data, using genetic imaging to do the prediction work, right? And then right now, like I mentioned, we could up to 30 million, at least 5 million SNPs there. How do we get the useful information as a feature to do the prediction? Right? I was just thinking about, if we're using, they mentioned this phenolist based on the SNP, overall important for all 1,000 phenols. that is one way to say, okay, not focused on one particular trait, but focused on overall trait. That's one thought I have. The other one is there any way we can find a like an indicator of the importance of the particular genome region? I'm trying to think, wrap my head around this. How do we leverage this information?
I think it will require sort of playing with the phenotypes a bit, because in the paper they clearly say, you know, here's the sort of overall distribution across the genome of associations generically. But really what we usually would want is what's the association across the genome for the psychiatric disorders or the cognition scores. And the psychiatric disorders, again, are, I think we looked at some of them, you know, there's not, because they are so aggregated, there's no, you know, one area where but there are some that seem to say, okay, you know, and then you'd have to spend some time drilling down to say, okay, what is it that's really contributing to that, you know, spike on chromosome 1? Is that anxiety, depression, schizophrenia? What is it? And they will let you do that. I mean, that it's actually, it would be an afternoon to really dig into that, I think. It's what might be worth doing.
Right. If we focus, I mean, that's good things about if we focus on psychiatry disorders and
or the cognition. I mean, they have a full cognitive, not a full cognitive battery, but a couple of cognitive tests that we could look at as well. I don't know if they, you know, get into attention per se, but they have some memory and other kinds of psychological assessments.
Yeah,
okay.
Chromosome 1, I know we used to have a favorite gene in chromosome 1.
Yeah.
Mir 137. No, they
Yeah,
have
but it's teeny, right? Isn't it buried
disability also in chromosome 1.
Isn't Mir 137 sort of buried in the middle of another gene anyway?
Yeah, it's but the region is when there's cell deletion is also when you have a big deletion in that region is intellectual disability.
Yeah.
I review grants, you know, on that, and it's really, it's a chromosome 1 deletion close to that region or including that region. So, but I don't know that. Again, the caveat, you know, schizophrenia usually affects the younger population. When we are over 50, then you can think about age as being a factor for neurodegenerative disorders.
Exactly.
So you have to be, you know, careful how you compare, you know, your schizophrenia population with other, but they are good for anxiety and depression. I mean, I would say this will be really good for that. Obviously, it will not help. It can help you for your Huntington's work because they are older populations.
Yeah, exactly.
But not ADHD or any younger. So, but still, I think it's a fantastic, you know, resource for all of us.
Broadens. I mean, if you know, if we're studying ADHD or something and we say, okay, here's the genomic region that we, you know, we think is interesting, and then we go look at it and say, okay, oh, it's actually related to neurodegeneration, or you know, in the UK Biobank, it's related to cognition. Well, that's kind of cool, you know. So there's definitely, I think it helps broaden the interpretation of what the genome is doing, even if it's not exactly the same phenotype as what we're interested in.
Yeah, I agree. Yeah, that's a good way of searching, you know.
Yeah, it's worth at least thinking about it, you know. It may be irrelevant, but it's worth at least looking.
Yeah, that's definitely help to, and like I mentioned, the interpreted results or the potential effect could be right. Okay.
Yeah, and I think if you can actually select by the chromosome region. So for example, with our QTL on chromosome 11, if we know the base pair numbers, you can go in and see what it's related to. If anything, you know, which traits seem to show up. If you know the gene name, Mir 137 isn't the official gene name, but if you know what the exact gene name is, you can type it in and see what it's related to, etc. So it's kind of it's fun to explore that. And it
Yeah,
might be actually.
MIR137 is actually a gene name too. It's
Oh,
all
okay. I typed
together
in MIR
with
137 and
capital.
it said, I don't know what you're talking about. So I figured I thought it was like MIR, like
It's-
H MIR dash something.
Yeah, it's MIR137 host gene. So it's
Oh, that's it.
MIR137HG.
HG, that's what it is. And is there a dash in it somewhere?
No, it's all together.
Let me see if
And in
that finds it.
capitals, like all the human genes.
Hey, they found it. Let's see. Is there anything there? Now you got me wondering. I'm going to do a phenome-wide association.
You know why?
Selected.
Someone asked me to review a paper on mirtrons, which is mir in an intron, like MIR137, just recently. And I've been busy, so I didn't review it, but it was interesting to see that
I'm trying,
the
but of course,
people
then you have
having
this. Yeah.
papers on MIR137 because it's important for neurodevelopment.
Oh, very good.
Mm.
It's got something around 10 to the minus 4 for psychiatric problems. It's not a really exciting area, but you can do if you have a particular SNP, right? Like our MIR137 SNP, you can actually go in and say, of all the phenomes that were, or of all the phenotypes that were tested, what's the distribution of p-values, and we could see whether there's cognitive and other things related to that particular base pair. But anyway, I know we're coming to the end, and I did want to, make sure we've given everybody a chance to chat. Was there any other comments or questions? Okay.
MIR137 is only for schizophrenia, no bipolar.
Right, exactly. It's very specific, so it's going to be
So
washed out
it will be diluted when you get anxiety, depression, and all of that. So it's not,
Yeah, you really want to. I mean, there's 800 phenomes and phenotypes, and I have not looked at every possible psychiatric. For all I know, they have a schizophrenia, but it's just not well represented. But they may have other measures as well. I know that there are relationships to that a couple of people have looked at. Great. Marvelous. Alrighty, well, I'm going to stop recording then. And thank you all so much for coming. And our next one is next